Okay, so what I'm going to talk about today, I was kind of keeping in the title, in the theme of what we've been doing, but I want to go way beyond the biology of xenobots, and I really want to talk about kind of what I see as the future of this field going forward, and in particular, this notion of an agential material and the kind of engineering that we're going to be doing with those materials. And I'm pretty serious about this title. I think that a lot of ideas as far as how we're going to go forward here are going to relate to ways that we think about not necessarily creating or programming, but literally raising new beings with various types of theories that exist around how you should do that. So this is the kind of thing I often start with. The end game for biomedicine. So there's this idea that at someday we will have an anatomical compiler. That is, you should be able to sit in front of the computer, you should be able to draw the plant, animal, organ, biobot, whatever, in any configuration, whether it's ever existed on Earth before or not. So here's a nice three-headed flatworm. And if we knew what we were doing, this thing would compile that description into a set of stimuli that would be applied to cells to get them to build exactly whatever you want them to build. On the biomedical side, it's obvious. If we had something like this, all of these things would go away. Birth defects, traumatic injury, cancer, aging, degenerative disease, all of that hangs on being able to tell a group of cells what they should be doing. But what about synthetic morphology? How are we going to make new kinds of forms and functions? And I think what's really critical about understanding the true potential of these ideas is that this is not some kind of 3D printer that's going to micromanage the precision of cells. This is actually a communications device. It's a translator whose job it is to convert from your goals as the bioengineer to the goals of the system. And in our group, we have been studying one particular interface to the collective intelligence of cells. The idea being that groups of cells are a collective intelligence. They navigate anatomical space. They have various ways to get around the perturbations, and they store memories, and they do things that individual cells don't know. And what holds all of that together, among other modalities, is bioelectricity. So the ability of cells to form electrical networks that basically do exactly the same thing that they do in their brain, in your brain. They provide a set of policies and mechanisms by which individual neurons get together and create a creature that knows things and has goals and capabilities that the individual cells don't have. And so we've been making tools for 25 years now. We've been making molecular tools to enable us to read and write into this medium. I want to show you some examples of what it means for what communication with cells and groups of cells mean. And so let's just take a look at the cellular level. I'm going to play this video in a second, but the colors here are voltage. Okay, so this is a voltage-sensitive fluorescent dye that reveals the voltage state of cells. And what you're going to see is these are two adjacent frames here. This cell obviously has a different voltage than the rest of the tissue, but then it's going to come over and it's going to just a tiny bit. It's going to touch this thing, and as soon as it touches, bang! It now has a different voltage. It's been converted or hacked by these guys to now do something different. So watch. So here he comes. He's got a special, boom. The minute he touches, okay? Let's see if you guys can see that again. All it takes is one little contact, bang, and his now voltage state is converted, and then he comes back and rejoins the rest of this. And so all of this is part of some work by Patrick Millen to understand under what circumstances cells do their own thing versus join a collective and then do something else. So this is an example at the cellular level of exactly the kind of thing we hope to be able to modulate in the biomedical kinds of applications. We would like to be able to provide signals, brief signals, that then drastically change the physiological state and behavior of cells. Now, at the level of multicellular organisms, we do things like this. This is again just some voltage imaging so that you can see the kinds of dynamics, both fast and slow, that exist in these in collections of cells here. And this is some work by Pai, who's here, and also my grad student Sherry Au back in the day, where what we discovered was that there are particular bioelectrical states that are interpreted by the surrounding cells to build specific organs. So when we inject RNA, specifically for certain kinds of potassium channels, the surrounding cells, even if they are cells that normally would form a gut. So what you're looking at here is the side view of a tadpole. Here's the native eye, here's the mouth, here's the brain up here. We inject a bunch of cells that might be gut. They get the message and they build a very nice eye. Not only that, the cells that we do inject, these blue cells are the ones we injected, they actually recruit all these other guys. All these other cells here were never touched directly by us. This is secondary instruction. We told these cells there should be an eye here. The message was sufficiently persuasive for them to take over that bioelectrical state and furthermore impose it on its neighbors. Okay, so we didn't have to tell them how to do that. The material already does it. By the way, it resists. These other cells actually resist because there's a cancer suppression mechanism that says if your neighbor has some kind of weird voltage, you should try to normalize them. So there's actually a battle of worldviews here. Some of the cells are saying we're going to stay gut or skin or whatever. These cells are saying, no, we're going to make eye. And those patterns sort of battle it out, and eventually, one way or the other, there's a solution. So understanding under what circumstances the signals that we provide are going to be not only convincing to the cells that we hit, but also cause them to organize their neighbors to do something quite complex, like build an eye, is really of key importance here. And then we can do many other things. You know, here's our six-legged frog, so we can induce limbs and portions of the brain and various other things. So there are other examples. So any kind of a significant intelligence should have some sort of a memory. These anatomical outcomes, in other words, when I have a piece of a fragment here from a worm, is going to regenerate as something, usually the same one-headed worm. How does it do that? Turns out there's a bioelectrical gradient that says one head, one tail. We can now rewrite that gradient, and you get these nice two-headed worms. And as you continue to cut them in future rounds of amputation and regeneration, we find out that that memory is quite stable. They continue to form two-headed worms. We don't have to keep stimulating them. Once we get this tissue to buy into a pattern that is a two-headed pattern, it will maintain this on its own. So, like any good memory, it's rewritable, but it's long-term stable. And if we want, we can actually also set them back. And here you can see a little video of these guys. So the idea here with all of this stuff, we've seen this right from the beginning, long before we really got into talking about collective intelligence and so on. It's just the idea that you have to take into account what the material is going to do when you provide these signals. There are many things it might do. It could ignore it, it could change it, it could propagate it to other subunits, and very simple signals. I mean, we don't know how to make an eye or how to make a head. We send very simple high-level signals that just say build an eye here or build a head here. And like in any good cognitive system, all of that propagates down to hit the lower levels, the biochemistry, the gene expression, all of that stuff is taken care of. Our job is to talk to the highest level that we can find with the simplest signals that don't require us to micromanage all the stuff that goes underneath. And then we can find out, by doing that, we can find out that actually the hardware that interprets these signals is really plastic. You know, these planaria, so this one has a nice triangular head shape. You can ask this tissue to make heads that belong to other species. Again, without touching the DNA, as we haven't in these other examples that I just showed you. So they can make flat heads like this P. felina. They can make round heads like this S. mediterranea. About 100 to 150 million years of evolutionary distance between these guys and these guys. And again, no change to the hardware needed, but with different bioelectric dynamics, they can visit other attractors in that anatomical state space and build other heads belonging to species that normally live in these attractors. So, by the way, not just the head shape, but actually also the shape of the brain, the distribution of stem cells, just like these other species. Okay. So the plasticity is massive, and so this is typically the typical talk that I would give to a biomedical audience would be this idea that everything today is at the hardware level. All of these kinds of things are kind of bottom up in terms of genomic editing and pathway engineering and so on. And we now have this opportunity for a massive amount of new approaches, basically including behavior shaping, which I didn't even talk about here, but all kinds of triggers through the bioelectric and through other kinds of modalities that can communicate complex, high-level goals to the material and let the system propagate that down to the molecular components. By the way, just to note, this is exactly what your nervous system does, right? So when you wake up in the morning and you have high-level social, financial, whatever goals you may have, in order for you to get up and exert behavior to execute on those goals, those very abstract things have to eventually make potassium ions flow across your muscle membranes. So the whole point of this system is to transduce very abstract goal states into changes in chemistry. Right? It makes the ions, ultimately makes the ions dance across the membranes. And so, right, so that level of propagation and synthesis is really critical, and it was very ancient. Evolution sort of discovered this long ago. So, and so then I would point out that, okay, so now we have some AI tools, and could we use AI to help us discover these high-level interventions at different levels all the way across? So not just talking to the pathways as standard drug discovery seeks to do, but actually all the way up to the cells, tissues, organs, and so on. Okay. But now, what's interesting to us here today is this idea of morphoengineering. And the cool thing about that for robotics, in particular biorobotics, is that because life operates in this multi-scale competency architecture, where every level solves problems, has agendas, and so on in different spaces, that means that when we engineer with this material, we get the benefit of all kinds of things that we would otherwise have to implement ourselves. So sensing, amplification, discrimination, all of this kind of stuff. If you were building traditional robots, you would have to implement all this yourself. Here, the material already does all of this, and so now the question is, how do we take advantage of all of this? I think we can go way beyond this kind of focus on the hardware to actually take advantage of the capabilities and the competencies of the material. And so we've talked many times before about our dream of building, and this is something that we're actually doing. Josh's lab and our lab together are actually doing this, building this kind of system that is a continuous discovery engine that flows in a cycle of AIs making hypotheses about how to communicate goals to these cellular collectives, actually building them. Right? So robot scientists actually build the Xenobots, applies whatever different kinds of stimuli that the AI wants, observes the outcome, modifies its hypotheses, learns from the experience, and does it again and again. So that's the kind of thing that we want to do. And so here I want to dive into this and really kind of talk about what it means to engineer with these kinds of materials. Because, and if anybody wants a lot of the details, there's a bunch of stuff here. But the idea is that we've gone way beyond here passive matter, such as some of the stuff that Thomas was talking about yesterday in terms of wood and metal and things. The thing about this stuff is that it has certain properties, and the tools that you have basically just assume that it's going to keep those properties. It stays where you put it, and that's basically it. And then we've had active matter and, of course, computational materials, but this is quite different. In biology, we deal with something that's quite different. And I think that in order to really take advantage of this field, we're going to have to break through some very ancient assumptions. So this is a piece of art called Adam Names the Animals in the Garden of Eden. And even for modern scientists, I think, consciously or not, this kind of thing really underlies a lot of our thinking, and it limits creativity. Because what's happening here, well, there's two things. One is I think deeply wrong and limiting, and then one is actually quite important for the future. The thing I think is wrong is that what this is trying to tell us is that there are discrete natural kinds. Okay, so here's a few different kinds of animals. We all know what they are. They're sort of discrete independent units. And Adam is of course different from them in some way, but it's this idea that whether it be whatever the origin might be, evolution has given us these very specific kinds of creatures in the environment. That I think has to go for reasons that we'll discuss. But what I think is super interesting about this is that in this original biblical story, it was very clear it was up to Adam to name the animals. God wasn't going to do it. The angels couldn't do it. It was really up to Adam to give them names. Now, in these kind of traditions, naming something means that you've discovered its true inner nature. Okay? And I think that's really fundamental here, because we are going to have to discover the true inner nature of lots of very weird and diverse creatures that we're going to be dealing with. And I think that will be important. Now, I really like to understand what we're trying to do here. I really like this example of the electromagnetic spectrum. So back in the day, before we had a proper theory of electromagnetism, we had static electricity, lightning, magnets, light. We had all these different things. And two things. First of all, we thought all these things were different. Okay? We thought they were fundamentally different from each other. We thought they were categorical differences. Nobody thought that they could in any way shade into each other. And if anybody's been in these debates that we now have in diverse intelligence, exactly the same thing. People love kind of sharp categories. So first of all, we thought these things were different, and second, we were completely blind to massive areas of the spectrum. We had no idea it was there. We couldn't take advantage of it because of our own evolutionary history. Okay? And what the advances in the understanding of electromagnetism allowed us to do is, first of all, significant unification to understand that all of these things are actually examples of one underlying dynamic. And two, it allowed us to create tools and to create a technology that exploits these other domains that we were completely oblivious to. And so I think this is exactly where we stand today with respect to diverse intelligence. We, first of all, the standard assumption is that there are real intelligent or cognitive agents, and then there's what they call dead matter. And so all of these things are different categories, and also because we as humans have a very specific and limited ability to recognize intelligence, we're pretty good with medium-sized objects moving at medium speeds through three-dimensional space, birds and mammals and things like that. We're okay with that. But anything at different scales or in other problem spaces, we find it very hard to visualize that that's an example of the same kind of thing that we are. And so I think this is significant mind blindness, and I think we're going to have to develop theory and then technology to overcome it exactly as we did here. There's one other interesting analogy that I like, which is the discovery of different kinds of numbers. So what happened, obviously first people had some counting numbers, and then somebody came up with zero, and then some negative numbers and some fractions, and then what's interesting is that every time this thing has been expanded, it was traumatic. It was painful. People got killed. This guy, Hippasus, actually got thrown off a ship for talking about these kinds of things. And it needs expanding the set of the set of things you understand under a particular category by softening the criteria and understanding that some of these criteria are just limits of our own imagination is tough, and it requires conceptual leaps. It breaks a lot of prior categories. And this is the kind of thing that I typically, when I give these talks, I go through a bunch of different types of creatures, including some very weird stuff that I'm not going to get to talking about today. I'm mostly going to talk about this and pointing out that we're basically going to have to expand a whole bunch of categories because the limits were artificial. I think they were never actually good, but they did okay. They served us reasonably well, but no longer. So this is the sort of thing that we want to talk about, is that this material is amazing. It self-assembles from a single cell slowly and gradually to one of these things, or even perhaps something like this. And lots of people make interesting claims about what the human mind is or is not capable of. And of course, there's never a story to be found around where and when exactly did that show up, either in the evolutionary sense or in the developmental sense. One thing you learn from developmental biology is that there is no bright magic line where you kind of go from physics to mind. That doesn't, there is no such place. It's a slow and gradual transformative process. So we need to understand this scale up. And then of course there's also all kinds of interesting things which I will talk about today as far as what can happen after this. This is not the end of the line for biology for sure. And so let's look at the material. This is the kind of stuff we're made of. So this is a single cell. So you can see this amazing structure that can be formed at the single cell level. But it's pretty static. It sort of does this, and this, by the way, to give you a sense of scale, this whole thing is one cell too. 5 centimeters. This whole thing can be up to 10 centimeters long. It's an alga called acetabularia, and it's got a stalk. It's got a foot. It's got this beautiful cap here. It has one nucleus. The whole thing is one cell. Right? So all the stories about different cells express different different genes, and they differentiate, and that's how you get a foot that's different from a hand. Yeah, this is all one cell. Okay? So there are tons of patterning kinds of mechanisms even within a single cell that can do a lot. And by the way, they don't have to be static. This is also one cell. This is a lacrimaria. There's no brain, there's no nervous system, but you can see it's got incredible. If you're a soft kind of a soft matter roboticist, you're probably drooling right now. We don't have anything that has this kind of control and plasticity and so on. So that's the sort of thing that we're made of. I want to show you another one that's pretty cool. This is called Physarum, the slime mold. I'm just going to stop it here. So what you've got going on here, this is a slime mold that is growing out of this piece here. It's sitting on some agar. All of this is a gel. And then you've got 3 little glass discs. These discs there's no food on them. They're completely inert. It's just glass. And for the first 600 to 700 minutes, this thing is growing out kind of in all directions. Okay, just by watching it, you're none the wiser in terms of what it's going to do. But it's actually doing something very interesting, and all that is described in the Roche's paper here. What it's actually doing is gently and rhythmically tugging on the agar. And when it tugs on the agar, it feels the strain angle of objects in its environment. And it remembers them and it integrates them. And so what it's doing is getting an internal information about things at a considerable distance. This is a plate that's about this big, okay, so considerable distance. And once it's integrated that information over a number of hours, what it's going to do is something very interesting, which is that it's going to grow towards the bigger mass. And it does this quite reliably. And so up until then, you had no idea where it was going to go. The growth is in all directions, but it's doing some kind of processing that we don't really understand yet. And you can play all kinds of games like stack these disks on top of each other. You can tilt the thing back and forth. You can put, we've done all this for the paper. It's really very interesting how it sort of pulls the environment biomechanically. And one thing you can see it doing, and by the way, again, one cell. This whole thing is just, it's one cell. What we did was inject fluorescent beads into this, and we're zooming in here to one little branch of this fractal structure. And so I'm just going to show you what happens. So first of all, you can see there's a lot of flow, okay? And so there's a branch here. And it kind of just looks like a passive garden hose at this point, that like, okay, you got a fork and everything's just going. But notice what happens here. It shuts off this branch. This thing is addressable at every branch point. So it's kind of a nice medium for some sort of hydraulic computing, because it has individual control of every branch point. It is not just a passive network. So even single cells have these amazing ways to get information about their environment, make decisions. I mean, we have no idea why it prefers the larger mass. So that's all cool, but then we find out that actually even the material inside the cells already has certain protocognitive properties. For example, gene regulatory networks or molecular pathways. Without needing the nucleus or the rest of the cell or any of that stuff, if you just have a very simple model of a few coupled ordinary differential equations describing how each chemical turns on or off some other chemical, what you find, if you're willing to test the various tools from behavioral science, you will find out that they're actually capable of 6 different kinds of learning. They can do habituation, sensitization, they can do Pavlovian conditioning, okay? And that's just a free gift from the math. You don't need to evolve anything, although certainly evolution optimizes the heck out of it. But these networks, actually just the networks alone are already, the material inside the cells is already capable of doing this. And as of a couple of days ago, Federico's paper is up, which shows that when you train these guys, actually their causal emergence metrics go up. Okay, so there's some I'm not going to pause now to dive into. People have questions, we can talk about it. There's some very interesting stuff that happens when you train these things. And in the lab now, we are trying to take advantage of that. So there are numerous applications for drug conditioning and various other things for biomedicine to try to either facilitate or wipe out the memories that the material inside the cells is actually forming. So what's happening in this multi-scale competency architecture is that every level is deforming the option space for the level below it, right? So what's happening is that as far as we can tell, that each level is so each level is just kind of the things that are doing what they normally do. They navigate their own space, but that space is bent and deformed such that the things they do are actually serving the goals of a larger next level up that these things may have no understanding of, and that kind of a thing And this is the plasticity of that material, the fact that it's hacking at every level. Every level is hacking the competencies below it, possibly upwards too, I don't know. Allows for some remarkable plasticity and capabilities here. So for example, this is Doug Blackiston's work where he basically prevented the two eyes from forming in this tadpole, but he put an eye on the tail. When you put an eye on the tail, it makes an optic nerve. The optic nerve goes out. Sometimes it synapses on the spinal cord. Sometimes it goes to the gut. Never goes to the actual brain. But if we use this machine that we built to test, to train and test them in behavioral visual assays, you find out that these animals can see perfectly well. So now this is crazy. Why is it that, the typical story you're told is, okay, you have some kind of sensory motor architecture, and it took millions of years of evolution to get things right. Why is it that in one generation, with no selection, no adaptation, no mutation, you drastically change the connectivity of this system? Yeah, no problem. It works. They can see the brain can use it. So that's one example of this remarkable kinds of plasticity. There are many other amazing examples, such as the fact that memory and behavioral information, or the product of learning, can actually move through the body. So these are planarian flatworms. You can train them to recognize these little bumpy areas as food, so it's called place conditioning. And so you train them to eat there, you chop off their heads. The tail does nothing for about 8 or 9 days. It just sits there until it grows back a new head. Once it grows back a new head, you'll find out that it actually remembers the original information. So not only are the memories stored outside, but at least partially outside the brain, but more importantly, they can be imprinted onto the new brain as it goes, because this animal does not have any behavior until the brain shows up. And you'll see, by the way, you'll see some planaria here today in Pi's section. But when the new brain shows up, it actually takes over that information from the rest of the body and then begins to act accordingly. So we're starting to see this amazing plasticity. And it's not just about holding on to the memories when the substrate is refactored. I mean, that's amazing enough. When you have these, this is actually some very old work by also Doug and some other people. When you have a caterpillar, you can train it, and then it goes through this process of basically breaking down its brain and then rebuilding a new brain that's suitable for the new world that it's going to inhabit. So a three-dimensional world as a hard-bodied creature, not a crawling kind of thing. Not only do these guys remember the original information, that's amazing enough. How do you store information when the whole medium is being taken apart and most of the cells are being killed off and so on? But there's a deeper issue here, which is that the actual memories of the butterfly are of no use to this caterpillar. The body's completely different. The goals are completely different. This caterpillar was trained to eat leaves when it sees a certain color. The butterfly doesn't want leaves. It could care less about leaves. It wants nectar. And the way it moves is not the same at all. So you can't just store the specific information. You have to generalize it, and you have to remap it onto a very different architecture. So what you're seeing here is that it's not only the plasticity of the body, it's not only the plasticity of behavior, but it's also the plasticity of the memories, the pattern memories that are moving across the substrate, not just from the tail end of a planarian onto a new brain, but to a radically different brain. Right? So this is the kind of stuff we need to understand. How are these patterns learned, maintained, remodeled, and sort of remapped onto different architectures? I think that's going to be a crucial part of this field going forward. And the other thing that the material can do, which I've sort of touched on as this multi-scale cognitive system, is that there's some beautiful top-down control. So, example, we take here's an axolotl. This amphibian, what you can do is you can take a tail and graft it onto the flank back here, and what you'll find is that over time that tail remodels into a limb. Now think about what's happening here. The cells at the tip here are tail tip cells sitting at the end of the tail. Locally, they're completely fine. There's nothing wrong here, and yet they turn into fingers. All the molecular stuff needed to turn tail cells into cells that belong in a finger is being driven by the very high level discordance between the overall structure of the entire animal. There's nothing wrong locally, but there is something wrong globally because tails don't belong in the middle of the flank. Okay? So the idea here is that and we know that regeneration is some kind of error minimization scheme, but it isn't just about damage. It isn't just about regrowing the thing that you lost. It's actually about making sure that the new pattern matches some kind of set point that, that you can use to realize that, wait a minute, this whole thing is wrong. And in order to correct that error, I'm going to have to filter down and change some molecular stuff down here. Okay? So crossing levels of organization and getting, again, it's this thing I told you a few minutes ago, that the higher levels bend the option space for the lower levels. You know, you still have to get, you know, it's not magic, you still have to get the genes expressed in the cells to move around and all of that, and you have to kill off a bunch of cells in between the fingers. All of that still has to happen, but it's driven by this high-level discordance between what's going on and some kind of internal set point, partially bioelectrical, partially maybe other things, of what it's supposed to be. And it also gives rise to this amazing kind of problem solving in other spaces. So what I just showed you is problem solving in anatomical space, right? So figuring out, wait, this is, you know, my limbs are not where they're supposed to be. That's anatomical space. Here's a kind of an amazing example of physiological and transcriptional state space. So this is something that we discovered a little while ago. You take these planaria and you throw them in a solution of barium, okay, barium chloride. Barium is a non-specific potassium channel blocker. It prevents the cells from moving potassium around. Many cells are unhappy about this, the head cells in particular, because there's so many neurons. They get very sick, the heads explode. Over about 24 hours, their heads explode. But if you leave the rest of the body in the barium, within a couple weeks, and even shorter if you do certain other things, they suddenly grow a new head, and the new head doesn't care about barium at all. No problem. Now, how can that be? So we basically compared with the transcriptomes of these barium-adapted heads with normal heads, and we found that there's only a handful of genes that are differently expressed. So now think about what this means. I always imagine this is like being inside a nuclear reactor control room, things melting down. You got a lot of buttons. You don't have a lot of time for random search or even hill climbing search in this scenario. It's too fast. The cells don't divide very quickly. You can't just start flipping switches. You are facing a problem you've never seen before. Planaria don't encounter barium. That's not the kind of thing that evolution would have prepared them for. And we've done this with other even more kind of exotic toxins. They have a physiological stressor, and they have roughly 20,000 potential effectors that they can fire off at any given moment, right? 20,000 genes. So that navigation of that state space. So you know, there was this concept of a Moravec bush, right? This robot, Hans Moravec, had this idea of a robot that had tens of thousands of effectors. And what kind of controller do you need to run a robot with tens of thousands of effectors? This is what's happening. You have to pick from that space. You have to decide, where do I navigate? Which of my effectors do I turn on to solve this problem? And they do. And we don't know how they do it. But this is the kind of thing we need to understand. We're dealing with a material that doesn't just hold its shape, but it propagates memories, it remaps memories onto new configurations of the material. It solves problems like this that it has never seen before in multiple different spaces, integrating across spaces. You have a physiological problem, you solve it in transcriptional space. This is the material is just incredible. We have basically no priors for dealing with stuff like this in engineering. It's a completely different thing. And I want to point out that we are not the only bioengineers. So this is, the typical thing that happens. Here's an acorn, it makes these oak leaves, and it's incredibly reliable. It happens, billions of times every year. You see these things, and we get this feeling that, well, this is what the oak genome encodes. Right? The oak genome knows how to do this, how to make this flat green thing. Except that along comes this non-human bioengineer, this little wasp, and the wasp can produce some prompts for the rest of the tissue to build something like this. These are called galls. If not for this thing, who would have even guessed that the oak genome, which normally builds flat green things like this, can actually build this? Okay? We'd have absolutely no idea that this is even within the possible latent space of this genome, but they do. And again, just to point out, these are not made from insect cells. These are made from the plant cells. They're just been the morphogenetic competencies of this thing have been hacked by whatever signals this thing. I mean, of course, it took this probably took millions of years. We would like to get there faster, but you get the idea. What I'm about to show you with xenobots and anthrobots and all that, it isn't unique to animal cells. Plants will do it too. And just all of this material has these amazing properties of problem solving and and error minimization, and it's hackable. It's because any system that has that level of plasticity, of course, is hackable. Now, one fun thing for us to remember is that it looks like the competency of the sophistication of the results that you can get is actually a function of the intelligence of the hacker. So when they make galls, bacteria basically make these kind of boring overgrowth blobs. Fungus does a little better. Nematodes kind of make this thing. The mites are making a little something a little more impressive. By the time you get to insects, you get something beautiful like this. Right? So there's some sort of relationship between the sophistication of what you can make the material do and what you're able to do yourself. And this, of course, is just one small snapshot. We really need to understand what's underlying this, so we can basically crank upwards on this axis. And so the idea is so this is why in developing our frameworks, I try to think about this from a very engineering perspective, with the idea being the axis of persuadability. That is, when we make claims such as these cells know XYZ, they remember something, they have goals, they have problem-solving capacities, whatever. I take all of those claims as interaction protocol claims. They're not metaphysical claims about what anything is or isn't. They are testable, empirically testable claims about, I'm going to take this bag of tools, maybe it's hardware rewiring, maybe it's cybernetics and control theory, maybe it's behavior science, maybe it's psychoanalysis, whatever it is. Here's a bag of tools, I'm going to apply it to the system, and we all get to find out how that goes, and then we can see, was it appropriate or was it a waste of time, and so on. And so I think this is really important. A lot of people have debates about this, about cells and tissues doing whatever, and a lot of those debates are basically armchair sort of philosophy about category errors and things like that. I think all of that is a waste of time. We need to simply take tools from other fields and see what, in fact, they give us or don't. And so this is kind of cool. As of a little while ago, so I'm going to so this is a part of the talk that's about xenobots and so on. And what I realized recently is that there's actually a movie being made in Mexico about xenobots. He's a superhero. Yeah, kind of amazing. But they got something deeply wrong, which is, if you read this, they're usually the typical thing. It's DNA altered. And I was like, oh man, they missed the most important thing. It is not DNA altered. Like that's the most important thing. But whatever, you take what you can get. We'll see. I think it'll be interesting regardless, and maybe we'll all go see it and yell at the screen when the DNA mutations start coming out. So xenobots. So the thing about xenobots is that they were specifically not genetically edited. We are engineering them by facilitating what I'm going to refer to, and we can get more and more into this. What we're doing is releasing constraints. So normally these cells up here, this is a cross section from a frog embryo. Normally these cells are basically forced by signals from other cells, so they are being hacked by the other cells in the embryo. They are forced to have a pretty boring life as a two-dimensional outer layer of a frog embryo that just keeps out the bacteria eventually of the frog itself. When you remove all of these cells, we're not adding any scaffolds or nanomaterials or synthetic biology circuits. We don't do any of that. We remove all these other influences, and we get to find out, what did these cells actually want to do other than what these cells make them do? And there's many things they could do. They could die, they could crawl off and leave each other, they could form a nice flat monolayer and culture, many things they could do. But actually, what they do, and I'm sure you've seen this video 100 times, they come together, and I want to show you, this is what it looks like in close-up. So each one of these, hold on, oops, just a second. Yeah, each one of these things is a single cell. So we're really like magnifying on a little tiny area. Each one, this is a chunk of cells. This is another chunk of cells. I think it's incredibly amusing that this little thing kind of looks like a horse. And so you'll see what happens. It moves around in some way. It looks like a little puppet. Like we still don't really know how the movement works. It's sort of sniffing around over here. It looks like, and then there's going to be some sort of calcium event that's happening. So all of these cells, and if you watch these, I mean, it's like you could watch these for hours. There's all kinds of cool shapes swinging around and turning around and doing things. So these have been called xenobits or microbots or different things. But eventually they kind of come together, and when they do come together, you get things like this. So here it is using its cilia to propel through the water. They can go in circles like this. These are all videos that Doug took. They can kind of go back and forth like this. You can make them into crazy shapes like these, you can punch out the middle and make like a donut that will swim along. Here are some collective behaviors. So some of these are just hanging out. This one's going in a circle. These two are interacting. This one is going on some sort of very, very long journey here. So there's lots of different motion types. Here it is traversing a maze. The water is not moving. There's no gradient here. So it takes the corner without bumping into the opposite wall. And then at this point, it sort of spontaneously turns around and goes back where it came from. And so this is the sort of thing where we would love to have had some sort of calcium imaging, for example, to say what is going on inside that thing when it changes behaviors from this to that, right? And that's something for the future to really, and it's actually very challenging technologically. It's very challenging to do that, but we'll figure it out. So what was discovered a couple of years ago, and this had a lot of input from basically the predictions of the AI that was done by Sam Friedman and Josh's lab, is that these guys actually, while they can't do the kind of reproduction that frogs normally do, because they don't have any of the components, what they can do is the kind of thing that von Neumann wanted, which was a robot that goes around and makes copies of itself from parts it finds in the environment. We call this kinematic replication. So this dust out here, these are more epithelial cells, just loose cells. And what the bots do, either collectively or individually, is to corral the stuff into little balls. And because it's dealing with an agential material, as were we when we made the first bots, these little balls mature into the next generation of bots. And so guess what they do? They run around and they do the same thing and they make the next generation. And so you get these multiple generations. Now, as far as we can tell, this does not have a strong heredity. So it's not like the bots that each one makes are more similar to it than it is to others. So it's not, this is not the full reproductive cycle that you need for evolution. Nevertheless, they're making copies of themselves in a way that nobody had seen coming. And there are many other interesting properties. So this is Pai's recent paper, which is quite amazing. We asked the question, typically when you look at a creature and you're asking what genes does it express, there's some particular history, evolutionary history that you lean on that kind of makes sense with that history. So we simply asked, well, xenobots have never been here before. What does the transcriptome of a xenobot look like? So you can see here there are some hundreds of genes that are expressed differently in the bots than in the age-matched tissues. We analyzed this, many interesting things. I'm just going to focus on one thing, which is this cluster of genes related to perception of sound and mechanical stimuli. And we thought that's interesting. Could it possibly be that they're trying to that they're picking things out of their genome that will allow them to do new things in their lifestyle, for example, to react to stimuli or vibration? And so we did this experiment. Pai put a speaker under the dish, fed a certain frequency, and observed that prior to the vibration, they were doing this. As the speaker turns on, their motility changes quite radically, and then they go back almost to almost the same. So they certainly do react. As we saw, immotile xenobots and embryos don't actually do this. So this is quite a specific bot thing, and you can see in this paper many other interesting genes get turned on. So we're starting to see this idea that the material has lots and lots of surprises for us, that trying to micromanage it the way we have done in the past leaves almost everything of interesting on the table. We have to do way better than that. And we can see, as far as future applications, I mean, obviously we're going to want to try to control the shapes, the behaviors. Here's an interesting, we talked about yesterday when Thomas was talking, we talked about the calcium waves. So here's this one. This is Patrick's experiment where he pokes this guy, and okay, there's a calcium wave here. But within some number of seconds, these two guys, which are just sitting in water, they're not physically touching each other, they're sitting in this medium, they're not touching each other, they find out what happened to them. So it gives the option, the material already communicates across individuals. So that means that if you wanted some sort of a swarm behavior where they see something and then they tell everybody else, this is the sort of thing. We didn't have to engineer them. We didn't have to put in all the different layers that you normally would have to put in to sense something and then amplify it and then propagate it to everybody else. They already do this. So the next domain is some of the stuff that Pi is doing next, which is to understand the behavioral preferences and competencies of these guys. So what can they learn? What intrinsic preferences might they have? How much problem solving can they actually do? We don't know. But one thing that I think is really a really interesting avenue for the future is to use the material as a kind of agential sensor. So in other words, they no doubt know things about their environment that we not only don't know, but we don't even know how to ask the question. We don't know what to measure. So it's entirely possible that we create some kind of a amplification and probably AI-based interpretation pipeline where the front end of the sensor is not some passive mechanical sensor or electrical sensor. It's actually the bot. And you get to, if we can understand how do we interpret the physiological states of the bot, we can ask the material, we can ask it, what do you think of the environment that you're in, right? And find out many things that otherwise we wouldn't even know. So the coarse graining, the way that this thing divides up the world and tries to represent the properties of the outside world in its own internal states might be very different to how we're doing it. And I think we're going to learn a lot if we can learn to crack that. So now a couple of things as I finish up. One of the things that happened when we published all the Xenobot things is that some people said, well, look, you took them from an embryonic frog, and also we know amphibia are plastic. Maybe this is just a frog thing. They call them animal caps. This tissue that we take apart is called an animal cap. It's been called that for decades. Maybe it's just a frog thing. So I wanted to ask, like, okay, what's the furthest you can get from an embryonic frog? How about an adult human? And so what you're looking at here, and I sometimes give this talk by starting off with this video, and I just ask people, what do you think this is? What are you looking at? And some people say, well, it's some kind of thing you got off the bottom of a pond somewhere. It's a primitive organism. I say, okay, so what would you expect the genome to look like? And say, well, the genome has to look like one of these primitive genomes. And I say, good, if you sequence that thing, it's Homo sapiens. It's 100% human genome. Because these bots are made from explanted tracheal epithelial cells from human patients. And they make this little motile construct that looks like it looks and acts like no stage of human development. Now, something else interesting that I just want to point out here, historically, is that people went really pretty crazy around the time of the Xenobots. And by the time this rolled around, I was really concerned that when we published this, I mean, these are biobots made of human cells. I mean, I thought, like, if they were this worked out over frog skin, I thought they were going to go nuts over the human cells. But what I actually found out is that really didn't happen, and I think it's because the Xenobots really already broke a certain kind of barrier. It was already 3 or 4 years of Xenobots by the time the Anthrobots came out, and it just wasn't as shocking to people anymore. And I find that completely amazing. And I will also say that I receive numerous emails from people who either think they have been injected with Xenobots somehow, or they want to be injected with Xenobots. Either way, both completely untenable and impossible. However, I've had 0 inquiries from people who think they've been injected with Anthrobots, which is really weird to me because that actually could happen and actually will happen when these are a therapeutic modality. Like that's potentially at least possible. Nobody's worried about this, but there are whole communities online of people talking about how they know they have Xenobots in their bloodstream and so I think it's quite amazing the way that the Xenobots actually grab people's attention and sort of change the landscape so that after that, like human biobot robots, not as shocking anymore. I just thought that was amazing. And so here's a little Easter egg for Josh here, in that this is the protocol by which we make these things. But notice something interesting, that the patient may or may not be alive at the time that these things are running around. So we do get them from biopsy samples from patients that are alive, but sometimes not. And sometimes what you get is you get them post-mortem, and it's a weird kind of secondary life. The human is no longer there, but there is something, and it's sort of beyond even the HeLa. I mean, people have talked about the HeLa cell line that goes on after Henrietta Lacks had died and so on. But you can sort of imagine the different types of different types of living organisms that can be made to go on after the original embodiment has gone. Now, these anthrobots also have some remarkable properties. I'll just name a few. First of all, in their transcriptome, 9,000 genes different, about half the genome different than the tissue of origin. A massively different transcriptome. Lots of interesting stuff here. For example, they express a bunch of embryonic genes. So in some sense, the tissue starts to resemble an embryo. What they don't express is any Yamanaka factors. They do not express any of this reprogramming stuff that people are trying to do. Nevertheless, they're younger than the tissue they came from. Okay, so let me say that again. The anthrobots are actually younger than the tissue they came from. There's an age reversal, and the way we know that is through Steve Havart's epigenetic clock. So we send him the samples, and they have this very accurate human clock where they can tell here's the age of the cells you started with here, okay, and so here's the age of the actual anthrobots, and then they age, and then they die eventually. So it's kind of remarkable, and my guess is that this has a lot to do with the fact that their priors are saying, you're pretty old, you know, your telomeres might be short, you know, you've been around for a while, but everything around you screams embryo. You know, you're being bent into a pretzel, you know, there's other cells next to you that are expressing embryonic genes. There are no other tissues hanging around. Maybe you're an embryo. Maybe you should revise your estimate of how old you are, you know? So I think that kind of thing is a whole other approach, and this is, of course, part of our strategy on rejuvenation and whatever. But again, the material is remarkable in these kinds of things. You know, they self-heal. So if you do mechanical damage, pretty quickly they will. This is about less than 10 minutes. They kind of heal themselves. And they have this amazing property, which is that you know, they have a bunch of behaviors. So you can draw this epigram of different behavior types. They can sort of traverse scratch wounds. And when they land in those scratch wounds, they assemble into something. I don't know if the movie, you know, maybe the sequel will have anthrobots and then there'll be a superbot and whatever. But this is they can assemble into this little cluster, and what they start to do is to heal across the gap. So these are all human neurons that were plated here. We took a scalpel, made a big scratch like this, and they sit there and they start to heal. And you can see this is what it looks like when you sort of lift them up. This is what it looks like. And again, we didn't have to teach them to do that. They do this on their own. We can try to behavior shape it in ways that help us, but this is something they were already doing. Who would have thought that your tracheal cells that sit there quietly dealing with, you know, mucus and air particles have the ability to be an autonomous little creature that runs around, expresses thousands of genes in a novel way, and can heal your neurons? And by the way, this is the first thing we looked for. This is not experiment number, you know, 89 out of 1,000 things we tried. This is the first thing we tried. So who knows what else these things can do? And I'm sure the same is true of Xenobots. It's just, all of this is just like scraping the top of the iceberg. So I'm just going to close up with a couple of quick thoughts. Much like you know, with the Anthrobot case, we have to ask ourselves, like, here's a normal developmental sequence. This is what people would have said the frog genome knows how to do. It makes this, and it makes this, and it makes these little tadpoles. And so we know when the computations were done to do all of this, they were done in the evolutionary history of the frog, millions of years of the material bumping up against the environment to do the computations to get all of this right. When were these computations made? There's never been any Xenobots. There's never been any selection to be a good Xenobot. This is what they start out with. The exact same material can do this. This is that 80-day-old bot that I was talking about. It's turning into something. I have no idea what it's turning into. That this developmental sequence has never existed to our knowledge. Kinematic self-replication has never existed to our knowledge. So we really have to understand how to even approach the question of where do novel organisms' properties come from. Because we're going to need this kind of thinking in short order. Because not only is there a continuum developmentally and evolutionarily, but we all know, both technologically and biologically, that this standard human that features prominently in stories of philosophy of mind discussions and so on, is just one point on this continuum, and we're going to have all kinds of crazy novel architectures that we are going to live with. And the question is, okay, these spaces of form and behavior were shaped by evolution, great. What are the spaces and what are the attractors for these novel beings that we are going to live with? And so I'll just point out that one way that people go at this problem is to say, well, it's emergent. Basically, to me, all that means is we got surprised. You did some stuff and something happened that you didn't see coming. I'm not a big fan of that approach because it doesn't tell you what happens next. How do you find the next interesting thing? So what I prefer is kind of what the mathematicians do, at least some of them, is to understand that there is a space of patterns that affects the physical world, but isn't itself determined by the properties of the physical world. So here's this Halley plot. It's a plot of this simple little function in complex numbers. It has a very specific structure, and there is no story of physics or evolution or anything else that's going to tell you why it looks the way it looks. If you want to understand this, or if you want to understand why cicadas come out at 13 and 17 years, because these numbers are prime, that is not an explanation that comes from physics or from a history of selection. So I think that what we have to do is bite the bullet and say that whatever explanations we can get from evolution and physics does not tell the whole story. There is a latent space of patterns. These patterns are determined by whatever it is that determines the patterns in mathematics. And we can now use these synthetic morphology beings as vehicles to explore that space. They're interfaces or pointers into that space. And every time you make something, but also for cells and embryos and everything else, you are exploring patterns in the space, and we need to start understanding they're not just low agency patterns of mathematics, like facts about prime numbers. Some of these are high agency patterns that we would recognize as kinds of minds or behavioral propensities, and we need to start understanding. And so I think the actual Garden of Eden is going to look very, very different. And also, just to point out that it's not just that we are going to have some kind of one-to-one mapping between novel, strange kinds of hybrid bodies and whatever minds are ingressing through them. But if anybody's a fan of the classic Gödel, Escher, Bach, we have something going on here where the exact same material can actually the vocabulary even fails. I don't even know what the right way to say it. But the same, the exact same material actually has within it multiple ways of engaging with itself and with the outside world. And I think this notion of polycomputing that Josh and I have been working on is really important. And it means that this is not just that one static picture. There's actually a lot more going on here that we can't even begin to illustrate. And so in the future, for all of us, we are going to be living in a very different world. Everything that Darwin meant when he said endless forms most beautiful is like this tiny little corner of this space, because pretty much any combination of evolved material, engineered material, software, and these patterns that come from this space, they all form very diverse kinds of embodied minds. And we're talking cyborgs and hybrids and various chimeras. Some of these already exist. Many more are coming. We are going to have to understand how to have an ethical symbiosis with these beings. So I'm going to stop here and just summarize really quick. Biology uses this multi-scale competency architecture of nested problem solvers, and literally, we could do a couple hours on this, literally systems that creatively interpret the data that they have, meaning both genetic and physiological, in different ways to understand themselves in the outside world, which we can now start to at least a little bit model. We, I think, are going to be relating to these things via communication, collaboration, and other tools of behavioral science, not the kind of micromanagement that we've been doing with physics and chemistry. AI, of course, is a great tool for being part of this, but in particular, I think that we have to really move past the idea that we are going to force all these systems to do specific things, and really understand strategies that have been applied, of course, in theories of education and things like that, to how do you behavior shape intrinsic motivation. There are massive implications to this, and it goes way beyond biology, and it's a subject for another time. But the material, when you're dealing with this kind of material, you really have to understand the intrinsic motivations of the material. And this, I think, is the deep research program for decades to come, is to understand the mapping between the physical interfaces that we make when we make these plots and the patterns that are going to a form and behavior that are going to ingress through them. So I'm going to stop here and just thank the people who did all the work. And so obviously Doug and Pi, and the various other people who did some of the examples that I showed you earlier in the talk. And I want to thank lots of collaborators that we have. Of course, most of all, Josh and his group, but actually lots of other people. And you know, I have to do some disclosures. But for us today, the most relevant, of course, is Fauna Systems, that is our partner on the Zanabot side. So that's it. I'll stop here. Thank you.
I have a quick question. On the Horvath clock, when you measured the sort of the epigenetic age of the anthrobot, how long after? What was the timing relative to the actual body of the person?
Well, let's see. So what happens is they take the biopsy from the human, they freeze it immediately. Yeah. It stays in the freezer. We buy it at some point. We thaw them out about 3 weeks to expand the cells and make the first bots. So, you know, weeks.
Okay.
Yeah.
I'm always blown away by the healing, the neural scar results. Do you have any evidence that there is, like, you know, you get the bridge, you can see it. Is there functional integration happening over that bridge? Like, is there communication, or is it just, you know, strands of protein that, you know, have been gone across? Yeah. So we haven't yet studied, so we need to do that because I want to see when that kicks on. And I want to see in particular, I want the calcium responding bots and the calcium responding neurons, and I want to see them if they're talking to each other. Right? So we will do that. I haven't done that yet, but the only thing I'll say is that I can't tell you what kind of signaling there is, but those are not strands of protons. Those are bona fide neurons. The staining that we did, these are neurons. So it's not impossible that they're silent, but I wouldn't bet on that ever. Like, I'm pretty sure it's going to be functional. We just don't know what the communication is. Cool. Please. Go ahead, Randy.
In regular morphogenesis, do you understand when the initial patterns, whether bioelectric or otherwise, are established? Is that understood?
Yeah. Well, so let's say in the frog where it's the most accessible. So what you see is you get an unfertilized egg. That unfertilized egg already has a particular bioelectrical state. It is not simple. Eventually the sperm comes along, breaks some symmetries, and the pattern becomes more complex. But I don't think you can ever pinpoint a time before that happens. There is no time before that happens, because if we were to look, not that we have because it's technologically very difficult, if we were to look as the oocyte is being matured inside the frog, I would bet any amount of money that you would see patterns already there. So there is no time before.
Some questions. First of all, talking about the source, have you ever tried other alternative source, like, for instance, urine? Because
sorry, source of what?
Urine IPS. You can generate IPS from epithelial tubular cells that are lost daily by urine. It works. This is a group that Regina Villari has done in Vienna.
We have not. Yes, I mean that's certainly part of. You know, we need to be making these out of all kinds of.
Could be an easy way to harvest cells from in autologous fashion. That's the first question. Secondly, did you ever try a co-culture system or a diffusion chamber where you can maybe culture adults themselves, that they know the story of the organisms they grow within, and see whether direct contact or vibration, photonics, whatever, or simply the release of paracrine factors can affect in bystander fashion the behavior of these creatures.
Yeah, it's all of that is on the list. So.
And one more question is, when you have this neuronal rescue, is that the anthrobot talk to the surrounding cells and coax them into neurogenesis, or proliferation maintaining the morphogenic field, or part of the cells from the anthrobot become neurons as well?
We have no evidence for the anthrobot shedding cells that become neurons. I'm not going to say it's impossible, but we don't have any evidence of that. I think it's the bots inducing the neurogenesis.
Yeah. And if you tried cardiogenesis, is there any evidence they can transform?
We're doing that now. I don't have any data for you, but we're doing that now. We have cardiac scars and things like that. Yeah, we're doing that now.
Thank you.
Yeah.
You give us some really compelling examples of ways in which something like global memory seems to be stored more locally. Do you have any evidence or hypotheses about both how and at what level that's stored. So like, for example, the flatworm, you cut it in half, it's already been trained to have 2 heads, regrows the second head. How finely or how close to 1 head could you cut it for that to happen again?
Yeah. We don't know. We haven't done it. Those experiments are actually incredibly difficult, mostly because the planaria are very individualized. They all have it's crazy because they're all supposedly clones of each other, but they have individual behaviors. And so when you try to train them, it's like training humans, right? So you need they're tough experiments. So we haven't done it, but in theory we could. My guess is, and actually the substrate of memory in general that field is a mess because the old story of synapses is like cracking in many ways. I think that my own personal guess, for whatever it's worth, is that the living material is basically a multi-scale reservoir that all of it is being used. I don't think there is a single substrate of memory. I think the whole, basically what the nervous system is doing is it's serving as the front end of this crazy reservoir that it's learned to interpret, and that cytoskeleton, molecular, pathway, like all of it can be served as memory. That's my current guess. We don't know. We don't know what it is.
Yeah, thank you for the talk. I was wondering if you've observed any limits of biological morphospace. Do you think any structure can be built, or is there a limited set of possibilities?
That's a great question. So yeah, people ask that if is it are cells universal in some sense? Could they build anything? Obviously we don't know. Proving a negative is very hard. I think at this point we are, as a community, so bad at coaxing the material that I think any limits would be extremely premature. My gut feeling is that they are universal, and that if we knew how to communicate with them, any group of cells could build any structure, with one kind of exception. And so this relates to some stuff that Thomas was talking about yesterday. So, for example, the planarian cells have no enzymes that make hard tissue. So if you wanted something that had teeth or a horn or some kind of enamel or like some kind of hard tissue, I don't think you're going to get that out of planarian cells no matter what, because they just don't have the enzymes to make it. So I think there will be some limitations like that, but as far as shape is concerned, my bet is that if we knew how to communicate to them, they would make whatever shape you wanted. That's my guess. that's by and I'll just say that is absolutely not the mainstream opinion. So most developmental biologists like this thing called developmental constraints, and they will tell you that zebrafish cells can only, they should only make zebrafish and things like that. I don't buy that for a second, but we can't. It's too early to say whether they're really universal or not.
Mike, you had a slide, I don't know, it was first third, I think, you mentioned bending the option space at each layer. Can you just reiterate what you meant?
Sure. Let me just go back. So first the example. So the example is basically this. What you've got here is you've got this tail that we've surgically, we didn't do this. So this guy's group did it. You attach it to the flank of the animal. Okay. So as far as the entire collective of cells is concerned, that's an error. That's a mistake. But locally, these little cells here sitting at the tip of the tail, everything's fine for them. They don't have any error. Everything's great. They're tail tip cells sitting at the end of a tail. And yet they start to turn into fingers. Why do they turn into fingers? Because the higher level is able to transduce signals down that basically force all of the other, right? So even if you're a single cell, at your limited cognitive bicone of what's going on, everything's great, life is good. So you would never turn into fingers. So the other, what the larger level has to do is to deform your space of sensing, of reward, of error minimization, all these kinds of things has to be deformed. So you say, oh no, wait, we're supposed to be fingers, right? And so that's what's happening. So this is the map that I made to try to sort of visualize the whole stack, which is this idea that at any given level you've got components that do local things, and if those local things are ever to be in service of some larger goal, the higher level has to start bending their option space. So you'll start, there will be dynamics where you start getting some error signals even though there's nothing physically wrong. And we have a whole project on this. It's very interesting. There were these molecules that were originally used to signal the presence of, for example, heat damage, so protein misfolding and heat and things like that, and DNA damage and things like that. So those are problems on the level of a single cell. we're now finding that what evolution has done is reuse those to signal error on a much larger scale. Where there's nobody's poisoned, nobody's damaged, nothing is wrong with the cells, but the pattern is wrong, such as what I'm showing you here. And right, so what we're looking at is the propagation of error signals about things, about error relative to a large set point of which the lower level doesn't know anything about.
Thank you.
So one of the things I think that's super exciting about what you're doing right now is you're getting at this like fundamental problem in human psychology, which is how do we like agentically drive our behavior using goals, both in the near term and long term. And so I'm just curious how you're thinking about that in this work where you're moving from like these small organisms that can have goals that are pretty concrete into more abstracted goals, and how you're going to be able to measure that, and are they kind of biological frameworks for how these similar types of organisms that aren't humans represent goals?
Yeah. I mean, there needs to be, so at the first pass, what we're doing is we're taking everything we can scrape from behavioral and cognitive science and applying them in these cases. So that's fine. But ultimately, we have to close that loop and say, okay, but these things are also showing us things that we never realized, right, in behavioral science. And so that part is coming. I don't know what it will look like yet, but for example, the standard behaviorist criteria of habituation, association, like those kind of things, we've already seen when we apply those to the molecular networks. We see that some things match those categories perfectly, and that's great, and that makes it easy to publish the paper or whatever. But there's other things that are like, yeah, they're a little different, and so you can go one of two ways. You can say, okay, fine, then that's not habituation, or you can say, wait a minute, maybe our notion of habituation was a little rigid, and there's actually other ways to think about this, right? So there's going to be an interplay of those kinds of things, and yeah, that's the work of the next decades.
It seems like both methods and goals are stored in memories, and when you're looking at the different ways that they might be stored, I know that's early in the work, but are you trying to decipher like which is which, how those memories might be stored?
We are, and I didn't show you any of this stuff, but like for example, in the planarian regeneration work, that was like a huge part of it, is to look at the different bioelectrical states and try to say what are we looking at? Is this a static, make a head versus a tail here, or is this some kind of a? We don't know yet. That's still very much in development. But I just will say, in case I haven't said enough weird things today, that I think some of that stuff isn't. As I alluded to at the end, some of it isn't going to be found in the physical measurements anyway. So some of those things I think are going to end up being patterns, and we have some. It's too early to say anything, but we have some very interesting work looking at kind of a bidirectional interaction between an agent looking through a space of possible solutions when it tries to solve problems, and the patterns of the solutions themselves that are competing for the attention of the agent, right? So there's a very strange kind of bidirectional way to look at this that may or may not pan out. So
It seems like in the things that you've shown that's got to be true.
it seems like it, right? But that runs afoul of lots of standard assumptions in the field, and so we have to you have to go very slow with that stuff.
Yeah.
But
I'm going to ask you to speculate. So I was really intrigued, Mike, when you were talking about the millions of years of evolution that it took these organisms, or the cells in the organisms, to have this incredible plasticity. So then when you graft on these tail and then it becomes a flank, like that is a really complex process, but because it's developed for like on an evolutionary time scale, it knows how to do that very quickly. So I'm wondering, with your interventions and inventions really that are doing perhaps artificial selection and creating evolutionary pathways that nature herself has not shone a light on yet, how do you see like the longue durée of the evolution of biology when you start making more chimeras, like when you start like adding different cells, perhaps even with alien material like antibodies plus xenobots, like how are you thinking about long-term evolution that you're making like interventions in based on nature's ability to have this plasticity?
Yeah, well, yeah, that's, I can say just two things that may be relevant. One is that, you know, one of the things that, and I can send you, we just wrote like a proper discussion of this that I can send you, but in this example here, right, with this plank thing, in an important sense, this never happens in nature. This grafting kind of stuff doesn't happen.
Right.
This kind of stuff doesn't happen.
Yeah.
On the other hand, it happens all the time if you realize that because of the unreliability of the substrate, of the biological substrate, is really critical. Like when we build computers, we want, noise reduction kinds of things. We want abstraction layers that hide things, below whatever level you're working at. You want error correction codes. You want the data to stay where it is. You want the fidelity of the data. The biology can't do that.
Right.
And so instead, and I alluded to this when I was talking about the creative storytelling part, what the biology has to do at any given moment is tell the best story it can, given whatever information it has, with no allegiance to what it used to mean to anybody else. And so I think the reason that's for this incredible plasticity, I think the reason this stuff works is because every embryo never expected strongly for the eyes to be in the right place in the first place. Every instance of development is basically regeneration, because you've lost your whole body, you're down to one cell and you have to rebuild. And what are we building? We could build a human, we could build an anthrobot, like what are we building? And so I think that for the plasticity here is because it basically doesn't have strong, I mean, some like C. elegans might actually, right, be really committed to what it's going to be. But the vast majority of these things, I think they're doing the problem solving from scratch every single time.
Wow.
And so the limits of that creativity, I can't even begin to imagine.
Mike?
Yeah.
Just to the point earlier, I just have a theoretical thought. So the work you're doing, in contrast to Darwin's theory, I have a theory here, which is just like in a lot of mathematical frameworks, if you try to really derive a solution to a very complicated, let's say, partial differential equation, most of the time there are more than one answer, so there will be multiple. Okay. So what the Darwin theory will give us essentially is, in the evolution environment, given a constraint, and it happens to be one solution just fits into that constraint. But what you are giving here essentially is that lifting all that constraints, just finding out other potential solutions, and you are essentially giving more freedom and make more viable solutions essentially emerge. I think it's essentially you are creating a superset of what could or should have been there, in addition to what Darwin's theory essentially limits us into in a viable environment.
Yeah, I think that's very reasonable, but there's something else in addition going on here. So you know, the standard Darwinian story is quite compatible with the standard version of what they call phenotypic plasticity. So you have a plant, and if it grows in a dry climate, the roots look like this, and if it grows in a wet climate, the roots grow like this, and the leaves are different. There's a couple of different types that it makes. That part's fine. But the whole point of the standard Darwinian story is that there's great specificity between the history of interactions with the environment and whatever your current properties are. So it's what is the theory of evolution supposed to do? It's supposed to explain why this thing looks and acts the way it does, because it has a certain history. So when you have this radical break with that, when you say, okay, so we kind of know why the frog genome makes a frog, it's got this history, but also kinematic replication, which is never, like the specificity between that and the froggy environment is zero. You could have never said anything about that from the old environment. And so now, what's the predictive value of it, right? So I think it's even, I think what you said is true, but I think it's even worse than that. In other words, that it's one thing where you evolve something that has multiple outcomes that it can make, but when you lose the specificity between what happened before and what it's doing now, then I don't know, then we're kind of out to sea with what the whole theory was supposed to do in the first place. So I think maybe I probably better go, right? It's like way over something. I'll just ask me afterwards.
Thanks again, Mike.